As a grower, you are always working towards achieving the best quality of your crop. To make this possible, Eurofins Agro provides you all essential information related to water, fertilization, growing media, and plant health. In this webinar, Petra van Vliet explains the relationship between the composition of plant sap and plant dry matter.
Hello everybody, my name is Petra van Vliet and I work at Eurofins Agro, and I'm going to tell you something today about plant sap, plant dry matter and the relationship between the two. So let's get started. If you think about a plant consists of 90% of water and 10% of something else. 10%, and that 10% is 90% carbon and oxygen and 1% macro and micronutrients. So that's your nitrogen, your phosphorus, your magnesium, and your calcium. So if you recalculate it all and you think about your plant, 1% is your macro and micronutrients. So that's your magnesium, nitrogen, phosphorus, potassium, think of that. And those are the important nutrients because they determine your yield and they will determine your quality. These nutrients are important for your nutrient uptake. They are important for the production. They are important for the production of your crop. You need these nutrients to have a healthy crop, to get a good yield, and to get a good product quality. If one of these nutrients is lacking or is less available, it will limit your yield and your quality. That's the law of Justus Liebig. It's been years old and it's still valid. So if you want to know how it works with your nutrient uptake, you need to think about what nutrients do I supply in my drip water composition, so measure the nutrients there, or what is taken up by my crop. Measure the nutrients, for instance, in the plant, in the leaves. Nutrients are important because they have different functions. If you think about nitrogen, nitrogen is important for DNA, proteins, and plant growth. Very important for that. Iron is important for enzymes and is also a part of your chlorophyll. Cobalt, also very important for enzymes. Manganese, it's a micronutrient, but it's very important for your chlorophyll. You need to have it, otherwise your plant won't be green and won't be able to do the photosynthesis. Also, manganese is important for your crop resistance. Calcium, important for cell wall, cell divisions. And as last, potassium. Potassium makes sure that stomata in the leaf can open and close. So it's important, it regulates the transpiration of the plant. So these nutrients all have their functions, and it's therefore important that you supply them all in the correct amount. And then the plant needs to take it up. So this nutrient uptake depends, for instance, on plant characteristics. How many roots does it have? This is a genetic thing. It depends on the pH in the soil solution and in your substrate or your substrate solution, let's call it that way. Your EC is important. Climate is important for the growth of the crop. Temperature, light will determine how fast your crop will grow. Humidity is important for the transpiration and for the transport of nutrients in water. The physical characteristics of the substrate are also important. If you have lots of calcium and lots of phosphate there, they like each other. And therefore, the phosphate will be less available for plants to take up. Also, if you have clay in your soil, the clay will bind to the potassium, calcium, magnesium, sodium. That is also a stock from which nutrients can become available. It's an important characteristic of soil, the cation exchange capacity. Presence of microorganisms, also very important. Microorganisms will decompose organic matter. And by decomposing the organic matter, you get mineralization of nutrients. For instance, nitrogen, ammonium nitrate will become available and can be taken up by your plants. Also, microorganisms are very important for your plant resistance. And I hope you've heard that from an earlier session about soil life in soil and in substrate. I think there was an earlier webinar. Nutrients are also affecting each other in taking up. Antagonism and synergism. Potassium is taken up together with nitrate. So if you want to increase potassium uptake, you might add more nitrate, because a plant needs to be charged neutral. If a plus goes in, a plus needs to go out, or a minus, an anion needs to go in. But also, nutrients can affect each other negatively. That's an antagonism. If you have lots of ammonium, it will then limit the amount of potassium and calcium uptake. The pH in your substrate is very important. I already talked about that a little bit. What you see in this graph is your soil pH is on the x-axis, and the availability of the nutrient is on the y-axis. And you can see if the pH increases, it's the red line, the iron, manganese, copper, zinc, and aluminium decrease when the pH increases. When the pH decreases, so it goes to 4.5, aluminum becomes available, and aluminum is affecting root growth. You get stunted roots, you get less roots, you get a lower plant, and you get less yield. So your pH is very important. But you also can see your pH is important for your phosphorus. The highest phosphorus availability is about pH 6.5. There's one thing there what shows up is the calcium magnesium. It's the orange line, and it seems to be going up and up and up. That's not true. Above pH 7.5, calcium and magnesium will bind with the phosphorus and will thereby decreasing in availability. So the pH is very important that you keep that in the right ballpark. If you then think about nutrient uptake, there are 3 different mechanisms how roots can take up nutrients. The first one is the root just encounters the nutrient. Root interception is what it's called. The second way that nutrients are getting near the root is by mass flow. And mass flow is the nutrient is moving along with the water to the root and it's been taken up with the water. That's the second way, mass flow. The third way is diffusion. That means that the nutrient moves from a high concentration to a low concentration. Phosphorus is an example of that, but phosphorus moves very slow in soil. Potassium is another example of going from high concentration to low concentration. And the low concentration occurs near the root because the root is taken up this potassium. So you get a flow of potassium going from high concentration to near the root, as long as the root takes up the potassium. Once the nutrients are in the plant, you get transport in the plant. It's taken up in the xylem, and that means it's going together with the water. It's going from the roots to the leaves in the xylem. And that's partly organized because the leaf evaporates, transpirates water, so you get an underpressure of water, and water is pulled up through the plant to go to the root, to go to the leaf, sorry. So water goes from the root to the leaf because there's transpiration on top. In that water, nutrients are coming along as well. Then in the leaves, you have this photosynthesis process, so sugars are being formed. And the sugars, you have lots of high concentration sugars in the leaves, you have lower concentration of sugars somewhere else, so you get a flow from osmotic flow from high to low once again. And that's transport in the phloem. It's going from the leaves to all other parts in the plant. If you think about leaves and you want to know what's in there, there's 2 ways to analyze. The first way is a dry matter analysis. And that's an analysis of the total nutrient content in the leaf. So that's nutrients that are in the liquid, in the plant, they're in the cell membranes, they're in the enzymes, they're in the chlorophyll, everything. What it does, you put the leaf into acid and everything is destructed and you measure a total amount. and it's measured on an ICP-MS mass spectrometer. Your results are in milligrams or millimoles per kilogram dry matter. The second method is in plant sap analysis. And what a plant sap analysis does, it measures dissolved nutrients in the sap and in the cell vacuoles in the cytoplasm. And we measure that if you send in leaves or you send in material, it's frozen, and then it's formed and pressed. And the liquid that comes out of it, in that we measure the nutrients. The results are therefore in milligrams or millimoles per liter, because it's sap we're talking about. We're talking about a liquid. So if you want to do a plant sap analysis, you've got to make sure that the sample taking is done correctly. First of all, you got to think about, what do you want to know? Do you want to know for your whole area what the nutrient uptake is? For that, you need to take a representative sample. So you collect material at least 40 different locations in the greenhouse. So you for sure have a representative sample. If you now have an area in the glass house where it's growing bad and where it's growing very well, and you want to compare those two, make sure that you take enough sample from each of them so you can compare. Thing to do is collect the material always at the same time if you're thinking about doing measurements over time. Because what's happening with a plant is it evaporates water. So if you do, at the end of the day, there's a high chance that there's less water available in your plant. And plant sap is expressed per liter water. So we advise really the best time to take a sample is early in the morning when your plant has a high turgor, is very sturdy, there's not enough transpiration, and there's a lot of water available in the plant. And if you do it always at the same time, it's good to compare. The other thing you want to do, and I will come to that later, why that is handy, separate leaves into the lamina and the petiole. The lamina is kind of the leaf part, and the petiole is the little thing where the leaf is connected to, that connects the leaf to the plant. The petiole transmits the nutrients from the plant to the leaf. It's kind of a bus system in between. It's a transport system. And I'll tell you later on why that is important. Also, distinguish when you sample between old leaves and young leaves. The younger leaves are the more active leaves. They do the photosynthesis. They are on top of the plant. That's where most of the light comes in. So they are photosynthetic, very active. The older leaves are more at the bottom, and they are often seen as the stock of potassium, phosphorus, and magnesium. And they can be used if you run out of potassium. The potassium from the older leaf will then be transported to the younger leaf. That's why I'm calling it a stock. That's what the old leaf is for. After you have collected the samples, make sure that you put them in a closed bag, in a sealed, closed plastic bag, so there's no water evaporation, and you keep it cool. So there's no processes going on. Keep it cool, do not freeze it, we'll do that for you, but analyze it as quickly as possible. Keep it cool, keep it closed. Just limit transpiration, that's the main thing. This is very complicated. This is going to tell you, this table tells you how much material we need for plant sap analysis. And these tables are also available in all the protocols that you can find on the website. It will tell you exactly how much leaves and how much petioles and how much laminae you need to get a good analysis. So, for instance, if you do a cucumber, you need about 15 to 20 laminae, young leaves, to get a good sample. But these tables are in the protocols. They will tell you exactly how much you need for a good analysis. So what we've done some experiments. We have done, in 2019, a big experiment, and there we looked at cucumber, bell pepper, tomatoes, chrysanthemum, and strawberries. and we monitored the plant sap in these crops. And we looked at them, is there an effect of leaf age, young versus old? Is there an effect of plant part, petiole versus lamina? And also, if we have plant sap, how does that relate to the dry matter? These two different measurements, do they relate to each other? So let's have a look at some results. And there's plenty of results, but I'm only going to tell you something about the tomatoes. And for the tomatoes, we had 3 varieties, the Trovanzo, the Brioso, and the Sweetel. 3 different tomatoes, and the good thing is, you cannot see in the data which one's which. So the variety itself is not that not important for tomatoes, I must add. Focus in this presentation is on potassium and calcium. And the focus is on these two because they differ in the way they get to the plant, and they differ in the way they are transported in the plant. Potassium, very mobile. Calcium, not mobile in the plant. And calcium has this other problem, challenge. It's only taken up by young roots, so by newly formed roots. That's a challenge. And I'll tell you later on why. If we then look at some results for potassium in tomato, what you see here on the left is the plant sap, on the right is the dry matter. If you then look within the graph, the 2 bars on the left are the laminae, so that's the part where the photosynthesis takes place, and the 2 on the right are the petioles, so these are these transport bridges that move the nutrients from the stem to the leaf. And you see already that for the tomato, that we find more potassium in these petioles than we find in the laminae. The Y and the O, they stand for young and old. And there's a little bit of a difference in there. It's not significant. You see a similar pattern for the dry matter analysis. So that's in millimoles. Look at the difference in unit. Plant sap is sap, millimoles per liter. Dry matter analysis, millimoles per kilogram. Dry matter. Same pattern. Lots of calcium available in the petioles, in the young and old, and much less available in the laminae. Calcium shows a different pattern. Calcium on the left, once again, the plant sap, on the right, the dry matter analysis. And what you can see here is that in the laminae, you find a lot more calcium than in the petioles this time. So it's completely the opposite of what we find with potassium. If you look at the plant sap, lots more calcium available in the older leaf than in the younger leaf. And that's there, it's been put there. The leaf has been around for a longer time than a younger leaf. Therefore, it's not uncommon, it's not unlogical that there's more calcium in there. But there's more calcium in the laminae than in the petioles. You see it in the plant sap, you see it in the dry matter. If we then put these two together and we compare the dry matter with the plant sap analysis, and we do this for potassium and we do this for calcium, then you get the following patterns. On the left, potassium in tomato. On the x-axis, see it, dry matter. On the y-axis, the plant sap. The line that you see, the blue line, is the 1-to-1. That means it's the same amount of dry matter as the same amount of plant sap, and the plant sap should always be the same or less than your dry matter, because your dry matter is your plant sap plus what's already built in. What you see with the potassium is it's one big heap. The dry matter is almost similar to the plant sap. It's a very mobile nutrient. It's hardly built in, is what you can conclude. But you can also see there's no difference between old leaf and young leaf. It's a big mess. Not a big mess, it's mixed up, let's say it that way. Sounds better. If you look at the calcium, we certainly see a pattern there. First of all, there's a lot more calcium in the dry matter than in the plant sop. And there's a huge difference between the young laminae and the older laminae. They're really separated from each other. So the calcium works completely different than the potassium in the tomato. To conclude, there's a lot more, but there's no time. Nutrient uptake differs by crop. I haven't shown that, but that's for sure the case. The tomato works different than the cucumber. If we now look at the tomato, for instance, there's differences between old leaf and young leaf. The old leaf contained a lot more calcium than the younger leaf. If we, for instance, take chrysanthemum, we didn't see any difference between the old leaf and the young leaf for calcium. So therefore, the nutrient uptake differs by crop. There's also a difference between petioles and laminae. The potassium was a lot higher in the petioles for the tomato than the laminae, and the calcium was the other way around. And also, if you look at the relationships between dry matter and plant sop, you see differences. For tomato, you can say that the plant sop is almost similar to the dry matter, which means that the potassium is hardly built in, but is always mobile in the plant. It can be moved from top to bottom and from bottom to top. It's not built in. While the calcium, the plant sop is a lot less than the dry matter content. So what to do? Which analysis? If you want to think about, I want to know something about my crop, what analysis should I do? Plant sop analysis measures the most recent uptake of nutrients. It's a snapshot. It's at that time when you take the sample, that's what you measure, a snapshot measurement. It can be used, very good, for adjusting fertilization, because you know what the plant's been taken up. Young leaf, most active one. So if you need to think about what leaf should I analyze, and you don't have enough money, do the young leaf. It's the most active. But the old leaf is a stock and might therefore also be interesting to measure. But keep them separate. The dry matter analysis measures the recent uptake and what already has been fixed. It's been built in the plant. So it's snapshot plus history. The dry matter analysis is always seen as important for crop and product quality because this calcium binding, that calcium is important for your cell membranes, it's important for your cell walls, and gives you a firmer plant. So it's very important that calcium has been built in also. So that's what you can measure with the dry matter. If you do a plant sop analysis, you get a report in which there's 19 nutrients measured, and we give you some target values. And the target values are going to tell you, am I in the ballpark? Is it good or not good? Or is it less or is it too much? So then you can figure out what to do, and you may be able to adjust your solution, your nutrient solution. There's a lot more to tell. If you go to the websites that are on this page, you can find protocols, and you can get and also information on plant sop, on dry matter. There's also information on sending samples, how to do that. So visit the websites if you want to know more. And I'd like to thank you for your attention. And if you have any questions, mail horti@eurofins.com. Thank you.